Hello, my dear lovely students. A very warm welcome, guys, to this class. So, hope I am audible and visible. Everything is fine. A very warm welcome to all my students. As I have promised you that we will be coming up with a summary series that will be helpful for your 12th class examination. One thing, guys, you have to keep thing in your mind is your boards. They are super important. Either you are going for an interview or are appearing for any government test or the interview, wheresoever you will be going, you will be having your curriculum vitae with you. In that, the percentage of the class 10th and 12th matters. So we have to really work very hard, and our target should be high. And by understanding each and every topic, we will be going through the each and every line of the NCERT. And yes, I remember when last time I came for the summary series, many students they requested that there should be some questions also. So yes, today we'll be discussing some questions also, like how MCQs are asked, what about the long answer type question, what about the short answer type question. So these questions also we will be discussing. So hope you all are ready. If yes, great. Let's start. So let's start the session. This is your first session, and in the session we are starting with the chapter which is a human reproduction. So let's start one second. So today's target is human reproduction. Human reproduction. Very important. This chapter is many times long answer type question, not only the short. In fact, the most of the long answer type question you can expect. Means every year from this chapter one long answer type question surely comes. So definitely we'll be looking at the PYQs also. So let's get ready. Now first thing is process in human reproduction. Human reproduction is a process by which a baby is born, right? Starting from the process of gametogenesis, that is the formation of the sperms in the egg, to that of, once again, starting from the formation of egg and sperm to that of insemination, then the fertilization, then ultimately it will lead to the implantation, then gestation and parturition. Like this, there are lots of mechanism related to it. So first we'll be going through each one of these, because we are talking about the human reproduction. So definitely the topic is the sexual reproduction. So which topic is it? It is the sexual reproduction, not the asexual one. Sexual reproduction. Done. Now first it's given as gametogenesis. So what is a gametogenesis? Gametogenesis means the formation of gametes. It means formation of gametes, right? These gametes, they can be sperm, right? These gametes, they can be sperm. And if I am saying the formation of sperm, that means I am talking about the process of spermatogenesis. Spermatogenesis is a gametogenesis only. Spermatogenesis. So next is termed as means egg formation, which is termed as oogenesis. Oogenesis. Clear? So you all know oogenesis occur in case of female, and spermatogenesis occur in case of male. Done. Clear? No, nothing rocket science. Very easy. Now moving on to the next is insemination. So what is insemination? Insemination means deposition of sperm in female reproductive tract. So that means deposition of sperms in female reproductive tract, right, by male. This is insemination. Now next is fertilization. So what do we mean by fertilization? Fertilization means fusion. It means a fusion of male and female gamete. Clear? Now moving on to the next is, what is the next one? Next is the implantation. Implantation, that means the placement of, it means placement of blastocyst. So what's a blastocyst? Blastocyst is a stage of the, like, once after the fertilization we get a zygote further divides and ultimately what we get is a blastocyst in uterine wall. We will be discussing everything in uterine wall, right? Moving on to the next is gestation. So what do we mean by gestation? Let's have a look. Gestation means that period of 9 months. That means a period of time which a female keeps a baby in their uterus. Clear? Many students, they say it's a stomach. It's not a stomach, it's a uterus. The next is a parturition. So parturition means what? Parturition means after the gestation period, giving birth. Giving birth. So one thing you know, that we are the sexual, we show the process of sexual reproduction, we show internal fertilization, and other than internal fertilization, so the development is also internal. Ultimately we are viviparous, we give birth to young ones. So hope these points are clear. Now let's move on to the next topic. So first we are starting with the male reproductive system. So in male reproductive system, what all things are there? See, let's have a look. So in male reproductive system, what is present is, the first thing is the testes. Testes are there where the sperm formation occurs. Sperm formation occurs. Clear? Next thing is, you will find some glands are there. Some glands are present. So these glands directly, indirectly, it supports the sperm formation, which we'll see later on. Now the next, you know, other than the glands, what will you find? You will find some ducts are present. Ducts are present. Is this point clear? So all these three structures, they are present in male reproductive system. Now let's start and let's understand the flow of sperm. See, listen very carefully. Here the testes are present. Testes are present in scrotal sac. So see, testes. Testes are present in scrotal sac. Scrotal sac. Clear? Where it is present? Scrotal sac. A pair of testes is present in case of male. Where are these present? These are present in the scrotal sac, which is outside the abdominal cavity. Sperm formation occur here. So where, what does happens here? The sperm formation. What does happen here? The sperm formation. Here. Now from here, from testes, sperm travels, because ultimately sperm has to come out. So in the testis, scrotal inside the scrotal sac, the testes are there. Inside the testis, the sperm formation occur. So from testis, the sperm further travels into various ducts. It travels into various ducts. First, they enter into the retetestis. Retetestis. Then it enters into the vasa efferentia. I'll show you, don't worry, with the help of a diagram, everything. Then sperm enters into the next structure, which is epididymis. From epididymis, sperm goes into vasa deferens or vas deferens, one and the same thing. Then sperm goes into the next duct, that is the ED, that is the ejaculatory duct. Ejaculatory duct. Ejaculatory duct. From ejaculatory duct, it goes out through the urethra. Through the urethra. So all these structures, they are clear? Done. The testis, retetestis, vasa efferentia, epididymis, vas deferens, ejaculatory duct, and urethra. So this is the thing. Now let's have a look. Look at this diagram. Now first thing you will see here, the penis is present. So let's start with the testis. So here, this is a structure. This is a structure which is termed as a scrotum, right? Scrotum. Scrotum, the testes are present. A pair of testes, they are present. Can you see? Right? Testes are there. Clear? So from this testis, ultimately you will see the vas deferens is coming out. Otherwise, there are other ducts also, which I'll show you later on. Retetestis, vasa efferentia, epididymis are there. Now, this vas deferens further joins and form the ejaculatory duct. What does it form? The ejaculatory duct. From ejaculatory duct, ultimately, the urine also travels. The urine also comes in the ejaculatory duct. Ultimately, there will be opening of other glands like prostate gland, then the Cowper's gland, and ultimately the sperm enter into the urethra, and from urethra it goes out. Clear? Now, let's look at this diagram, and then I'll be explaining you each and every point in detail. So first, let's start with the testes. So the testes are situated outside the abdominal cavity. Where are these present? Outside the abdominal cavity. It is not present in abdominal cavity. It is present outside because it requires temperature which is around 2 to 2.5 degrees Celsius lesser temperature is required for the proper spermatogenesis to occur. Yes, when a baby is born, testes are present in abdominal region. Due to the action of testosterone, the testes, they descend into the scrotum. When they fail to descend into the scrotum, this is termed as cryptorchidism. It's a condition. For that, they have to go for a surgery where the testes, they should come out, right? So they take out the testes from the abdominal and they place them, doctors place them in the scrotal sac. If we'll not go for this surgery, the testes, because they are present in abdominal cavity, temperature is quite high. 2 to 2.5 degrees Celsius temperature is high. So it will not allow a proper spermatogenesis to occur. Clear? So everything is temperature regulated. Now, scrotum helps in maintaining the low temperature. Very important question. This has been asked in one of the MCQs. So get ready. 2 to 2.5 degrees Celsius lower than the internal body temperature necessary for the spermatogenesis. Moving on to the next, in adult, each testis is oval in shape, right? What was the shape? Let's come to this. See, what was the shape? It looked like a oval shape. Clear? It's a oval in shape. Very important. Length is 4 to 5 centimeter. A width of around 2 to 3 centimeter is there. It is having a dense covering. So, look at here. I'll make a diagram and I'll make you understand this topic. Now, if you look at the testis, an oval shaped structure is present. So here the two coverings are present. This is the covering number 1 and this is the red one, the white one is the covering number 2. So here this covering is termed as tunica vaginalis. Here they are saying some covering. So yes, so let's talk about these coverings name. Vaginalis. The internal covering, this is also termed, internal is termed as tunica albuginea. Tunica albuginea. Guys, are you getting these points? They are very easy, right? So hope your revision is going on. So here the internal layer, it forms of some pouches, right? Which are termed as testicular lobules. What are these? These are the testicular lobules. Testicular lobules, very important part, lobules. Now in testicular lobules, you will find the, I'll show you, wait, which color? Okay, we'll take this color. Here you will see the seminiferous tubules are present. Some tubules means some tube-like structure is present which is termed as seminiferous tubules. So I'm labeling this here. These are the seminiferous tubules. What will you find here? The seminiferous tubules. Seminiferous tubules are there. Clear? What do we find here? Seminiferous tubules. Now let's have a look. Now you know about the dimension. No need to remember the dimension. This is enough, right? Now the dense covering is there. We have talked about the dense covering, two covering, tunica vaginalis and tunica albuginea. One which is present towards the outer side is a vaginalis. Towards inner side, it is a albuginea layer is present. Testis, 250 compartments are there which are termed as testicular lobules, which are the extensions of the tunica albuginea. Clear? So how many? 250 testicular lobules they are present in each testis. Now come to this diagram. Now I'll show you. See, look at this very important diagram. Here you will see testicular lobules are present, right? So here testicular lobules are there. Now ultimately testicular lobules, see, I'll make a structure, right? I'll make a proper structure to make you understand the topic, because from this topic many times the question has been asked. So this is the tunica albuginea. The red one which we draw, that is the tunica. Yes. So this is the layer. Here, these are the compartments, and the name of these compartments are what? Testicular lobules. So let's label this here. What is this? Guys, come on. Everybody keep on saying testicular lobules. How many testicular lobules? 250 testicular lobules are there. How many? 250 testicular lobules. Clear? In each testis. Here, some like this structures are present which are termed as seminiferous tubules. Now earlier I was talking about the testis where the sperm formation occur. Now we are going deep into the structure of the testis. So here what I am drawing right now is the seminiferous tubules. Seminiferous tubules. Clear? Tubules, long tube-like structure. Now, listen very carefully. Now, I'm making the diagram very much simple for you. Very much simple for you. Here, these are. Can you see this structure? Now, I am making the diagram super duper easy for you to make you understand the topic, guys. If you will not be able to understand, that means you will be cramming things, and that I don't want. So here, this is how they opens. Clear? Now, the one which I have drawn in the green, what is this? This is the tubuli recti. Tubuli recti. Tubuli recti ultimately opens into this duct. What is this? This is termed as rete testis. Rete testis. Rete testis. Now, I am using now another color for the better understanding. See, from here, I'll use one more color. Wait. So, this is how the things look like. Here, let's label this. What is this which I have drawn in pink color? This is the vasa efferentia. This is the vasa efferentia. What is this? The vasa efferentia. Ultimately, it opens into, what is this? Which structure I have drawn? This is termed as, here. Guys, can you see? Which structure is it? See, I'll show you with the help of this diagram. Okay. If it is here. Yeah. Now, not given. Here. Guys, can you see this structure? This structure like this. This structure like this, right? A cap-like structure. So what is this? This is a epididymis. So ultimately it opens into epididymis. Epididymis. Right? Here the cauda part, right? So the cauda part, ultimately this one part opens into the vas deferens. So where does it open? It opens into the vas deferens. So this structure is vas deferens. This structure is vas deferens. Clear, buddies? Right? Easy, right? Easy peasy, lemon squeezy. Very easy question, right? So ultimately what will happen? See, vas deferens. This is a vas deferens going on. Now vas deferens will go up, and ultimately here it will form an ampulla-like structure. Right? It will form an ampulla-like structure. So what do we call this? Ampulla only. Ampulla. Here, a gland is present. This is termed as seminal vesicle. This is termed as seminal vesicle. Seminal vesicle ultimately opens here, and what they form is ED. That is the ejaculatory duct. That is the ejaculatory duct. Clear? Seminal vesicles, they secrete some substances which are important, which basically provide the nourishment, which I'll show you later on. The exact function of all of this. See, this is the actual diagram. See, this is a one testis from where the vas deferens is coming out. This is the vas deferens properly labeled. Here the ampulla-like structure, the bigger, a broader structure you can see. The ampulla-like structure is there, and ultimately you will see here here the seminal vesicles are present. A pair of seminal vesicles are present, which opens into the. Ultimately, what we will get is the ejaculatory duct. You can come to this diagram also. I'll show you here as well. Here, buddies, this is the vas deferens coming. If you look at the structure, this is the vas deferens coming. Here, the seminal vesicle are present, and this seminal vesicle ultimately opens into the ejaculatory duct. Clear? This opens into ejaculatory duct. Now, here comes the major structure, which is termed as the urinary bladder. Now, here, this is a urinary bladder. Ultimately, urinary bladder opens into this. Clear? Urinary bladder. This is a urinary bladder. Now we'll not call this as the ejaculatory duct. Here, further there will be opening of the prostate and the Cowper's gland. This is the Cowper's gland. Right? The another one is the prostate gland. Prostate gland. And finally, what do we get here is this. This is the urethra. So, urethra carries a sperm as well as the urine. Now, if you, this is of the one side story. This is a one side story. Like this, there will be another side story also. Same thing will be of other side story also. Clear? Now, here, the penis is present. So, ultimately, the structure look like this. See. So, this is how the penis. So, this is a penis present. Right? What is this? This is the urethra. This is a urethral opening. Urethral opening. Right? What is this? The swollen structure is the glans penis. This is the glans penis which is present. Here, one skin is present, which is termed as foreskin or prepuce. Foreskin and prepuce. Foreskin and prepuce. So, this is how the whole structure look like. Right? Now, here, if you remember, we talked about some seminiferous tubules are present. Now, if you will take a section of the seminal vesicle, if you will take a section of the seminiferous tubule, the structure somewhere or other look like this. Now, this one structure, guys, can you see? This is the structure, right? This is the one seminal vesicle. I'll use another color for the better understanding. Wait. So, this is the one which you can see. This is a seminal vesicle. I told you it's a duct-like structure. We have taken a transverse section, so it is appearing round. Here, the germinal epithelium is present, right? What will it find is germinal epithelium. Germinal epithelium. This is a PYQ. Like which structure, which type of cells are present here? This is the cuboidal. Cuboidal. Clear? Simple cuboidal epithelium is present here. So ultimately, they forms the sperm. Here, what they form is the sperm. Here you will see the cells are present in various stages of development. Ultimately, you will find the sperms over here in the center. Right? So these are on the periphery. You will see the immature sperms are there. Immature sperms. In the center, you will see mature sperms are there. Mature sperm. So, how does this development occur? I'll show you in the spermatogenesis thing. Clear? Now, in the center lumen, you will find the sperms are present. Now, here, different type of cells are present. Let's understand that. Here, some special type of cells are present. These special type of cells, they are termed as Sertoli cells. Sertoli cells, right? Sertoli cells. So Sertoli cells are those cells whose function is to provide the nutrition. What do? They provide the nutrition to the developing sperms. Developing sperms, they are going to provide the nutrition. Now, some cells are present over here. So these are termed as interstitial or Leydig cell. Interstitial or Leydig cell. Another name is Leydig cells. Clear? Here, these are present. Now these Leydig cells, these cells which are present over here, these cells, they secrete androgen. Now out of various androgen, one which is given in your NCERT is the testosterone. Testosterone. And testosterone is the one which is important for the secondary sexual characteristics. Secondary sexual characteristic. As well as this is important not only for the secondary sexual characteristics, this is also important for the spermatogenesis. For the spermatogenesis. So secondary sexual characteristics means they men, they are having low pitch voice. Females are having high pitch voice. The low pitch voice, all thanks to the testosterone. Now the body here, the pubic here, development of mustaches, development of beard, that is all because of the testosterone, right? Clear? So this is how the sperm formation occurs. So sperm formation starts at puberty, but it is a lifelong process. Throughout the life of a male, sperm keep on forming. Done? Clear? Now, let's do a quick NCERT reading. The male sex accessory ducts include retetestis, vasa efferentia, epididymis, and vas deferens. These are the ducts present. Now, what else is? The seminiferous tubules of the testis open into a vas efferentia through retetestis. The vasa efferentia leaves the testis and open into epididymis located along the posterior surface of each testis. Epididymis leads to the vas deferens that ascend in the abdomen and loops over the urinary bladder. Clear? Is this point done? Now, remember very, very, very carefully. Like I told you, now this helps the formation of the testosterone. Now this provides nutrition. So I'll show you how does it occur. When does the formation of sperm starts? Let's have a look. So please remember, at the time of puberty. At the time of puberty. Puberty. Clear? At the time of puberty, what happens is we are having hypothalamus. Hypothalamus sends signal to the anterior pituitary. Anterior pituitary. Anterior pituitary secretes two types of hormone, which is one is FSH and another is LH. Clear? Here, the hormone which is secreted by the hypothalamus is GnRH. Right, which is gonadotrophin releasing hormone. This you have done in the class 11th also. Gonadotrophin releasing hormone. Clear? Anterior pituitary cell secrete follicular stimulating hormone. What's the name? Name is follicular stimulating hormone. And LH. What is LH? LH is luteinizing hormone. Is luteinizing hormone. Follicular stimulating hormone and luteinizing hormone. This FSH go to Sertoli cells. Where does it go? It goes to Sertoli cells. Where are Sertoli cells present? Sertoli cells present in the seminiferous tubule. So what does it do? It provides nutrition to the developing sperm. Nutrition, right? It also secrete hormone which is inhibin. Inhibin. Clear? Now the luteinizing hormone goes to the Leydig cells. It goes to the Leydig cells. Clear? L for luteinizing hormone, L for Leydig cell. Leydig cell will help in the Leydig cell will secrete testosterone. Testosterone. So it goes, and here it causes the positive control. So this is the, it allows more and more synthesis of, whereas the inhibin does the negative control. Clear? So this is how the hormonal control look like. Now we have talked about some glands. See, come here, come to this diagram, come to this. What we were talking about is testis. Now testis is done, seminiferous tubules, various ducts are also done. Clear? Now these two topics they are done. We have to talk about the glands. Let's come to this now. Glands. So how many glands are there? C. Accessory genital glands. So there are three types of accessory genital gland: prostate, the seminal vesicle, bulbourethral or the Cowper's gland. First, let's talk about the seminal vesicle. First, we talked about the seminal vesicle, that this is the one which opens into the ultimately into the ejaculatory duct. How many? One pair is present, situated behind the urinary bladder. Right? What does it provide? Right? It provides basically fructose. Fructose is a sugar. And sugar is what? Sugar is important for the food. Right? It provides prostaglandin, right, for their maturity. This is important. Vitamin C, citric acid, and inositol. These are the things which are provided by the seminal vesicle. How many? Pair. Yeah. Now moving on to the next is the prostate. Prostate is a chestnut shaped, unpaired, provide milky appearance. So milky appearance is because of the prostate gland. They secrete calcium ion, proteolytic enzyme, citric acid, and seminal plasmin. See, whenever sperms are deposited in the female reproductive tract, it thickens itself. Later on, the semen thicken itself. Later on, the semen liquidify itself, and then the sperm they start moving in the female reproductive tract. So this is because of the seminal plasmin. So their consistency changes with because of the seminal plasmins. Clear? Rest all you know. Proteolytic enzymes, they are important for the penetration. Citric acid is important because it provides the acidity. Acidity is important. So they require a particular pH for their activity. Fructose, food, prostaglandin, movement. All these are super duper important. In case of a rape test, yes, for the rape test, fructose is seen in female reproductive tract. If fructose is present, so that means the sexual intercourse has done. Now, one pair, bulbourethra, secrete mucus and it helps in the lubrication. So all these are super important, guys. Now, let's move on to the structure, which is the penis. So penis is the male external genitalia. Clear? This is the direct line from the NCERT. Made up of special tissue which helps in the erection of penis to facilitate insemination. See, I'll explain you this. There is erection of penis. How does this erection happen? Because some special type of tissues are present in the penis region. So if you will take out a section here, the section is like this. Some corpus spongiosum, some tissues they are present. And here the urethra opening is there. So this is the corpus spongiosum. Spongiosum. Right? Here corpus cavernosum. And here this is the urethra. Because it's a section, so urethra looks like this. This is the urethra present. Clear? So here these tissues, corpus spongiosum and corpus cavernosum, these are the erectile tissues. So at the time of erection, at the time of copulation, now here some the ducts which are here, whenever the signal comes from the brain, so here the neuron over here, it causes the vasodilation. Blood vessels are there, so vasodilation will occur. Blood will come and deposit in the corpus spongiosum and the corpus cavernosum. Whenever they will deposit, it will lead to the erection of the penis. So specialized tissues are present. So enlarged part we have discussed, that is the glans penis is present, and here the covering is present on the glans penis, which is termed as the foreskin or the prepuce. Moving on to the next topic, what does it secrete? It secretes sperm. So secretes sperm. So sperm, you all know the secretion, ultimately the secretion, whatever comes out, that is termed as semen. So semen is having two different things, that is the sperms are present plus the secretion of secretion of glands. Gland secretions are there. Clear? So that is termed as semen. Now moving on to the next. What is this? It's a microscopic structure. Here the different structures are present. Let's have a look at the structure and then I'll make you understand the whole topic. Otherwise then the reading is not important. Now here, this particular portion, this is termed as a head. Second portion is a neck. Now this one is a middle piece. Then the tail region is present. How many parts? Here the 4 parts are given. Head, neck, middle piece and tail are present. Head, you will see that everything is covered by the plasma membrane. Plasma membrane, it's a normal cell only, but during the process of development, all these structures, they develop, the cytoplasm is reduced, Golgi body lead to the formation of a structure which is termed as acrosome. So acrosome is formed by the Golgi bodies. Golgi body. So here acrosome, here what do we have? What do they have is the acrosomal enzyme. Acrosomal enzyme. So what's the function of acrosomal enzyme? Acrosomal enzymes helps in easy penetration. What does it help? In the penetration, so that the penis can be so that the sperm, they can go into the egg for the fertilization. How does it occur? That I'll show you. So here the acrosomal enzymes are present which helps in the digestion. This is the nucleus. Chromosomal material is there. Now the nucleus which they are having, they are highly developed. In fact, the cytoplasm is not that developed. Here the next portion is the neck region. In the neck region, what all structures are there? In the neck region, you will see the proximal centriole. Proximal centriole is present and the distal centriole is present. Distal centriole. 2 centriole are there. This you have all must have done in the 11th class in the cell structure. Now from here, this is the axial filament that comes out. What is the, from the distal centriole? Axial filament, which further forms the tail. Here, if you look at the mitochondria look like this, so which is a source of energy for the easy swimming of sperm in the female reproductive tract. Done. Structure is done. Many times this question has been asked about the acrosome, the Golgi body. It is derived from the Golgi body. Hope it is clear. Now, very important part regarding the sperm. So, how many sperms that, like, what is the quantity of sperm which is being produced? Now, here till this point we have done all these points. I have explained you. This particular point, this is done, this is done, this is also done. I have explained you. Now, in human male, very important, 200 to 300 million sperms are present. So just imagine how many? Lots of, literally lots of. Clear? Out of this much during the coitus, at least in the one coitus, and you know, out of this 60% of the sperm, they should have a proper shape and the structure. 60% of the 200 to 300 million sperm, they should have a proper structure. Out of that 60%, 40% of the sperm should have the 40% vigorous motility. Many times the student, they get confused. that 60%, if the out of the all the sperms per ejaculate, 60% they are having proper structure, and out of the whole semen, if 40% is having the motility, no, it is not the case like this. Once again, it is not the case like this. In fact, guys, the out of the total sperms which are produced, 60% of that should have a proper structure, and 40% of that 60% should have a proper motility. Clear? And if this count is less than 20 million, so that means infertile. If this condition is fulfilling, that means 61% and the 40%, the person is fertile. So this is important if someone is going for any test. So at that particular time, per ejaculate, sperm count is done. Sperm structure in the structure is also calculated. Like out of all the sperms, how many percentage, in terms of percentage, how many sperms they are having a proper shape, how many are having a proper motility. So everything is noticed. Moving on to the next is a female reproductive tract. So this is how a female reproductive tract look like. But a better structure is like this. This is the structure of external genitalia. I'll explain you everything. Now, in case of female, right? In case of female, a pair of ovaries are present, like that of a male testis. We'll be doing everything comparison. A pair of testis is, a pair of ovaries are there. A pair of ovaries are there. Here also, buddy, some ducts are present. Ducts are present. And here the name of these ducts, they are the number 1, fallopian tube. Fallopian tube is there. Here, the next tongue is the uterus will also be present. The third is the cervix. Ultimately, the vagina are present. So this is the internal structure. A female reproductive structure is like this. If a female reproductive system structure you have to draw, this is the diagram, guys, you are going to draw. And when it is all about the external genitalia, diagram is something else. That I'll explain you that also. So just have some patience. Here you will see the ovaries are present. This is the ovary number 1. This is the ovary number 2. 2 ovaries are present. Now here, like this is the ovary. Let's say this is the ovary, and this is how the fallopian tube, the opening of the fallopian tube look like. They are not in touch with each other. Here the ovaries are attached with the help of a ligament, a pair of ovaries. Now this is a fallopian tube. This is a one fallopian tube, and this is another fallopian tube. Fallopian tube is having various parts: infundibulum, ampulla, and isthmus. So this starts with the infundibulum. Infundibulum is having some finger-like projections which are termed as fimbriae. Now the structure is like this. Here a hole is present which is termed as ostia. So the from the ovaries, eggs are released in the cavity, and later on the egg travels from the ostia and it goes into a pipe-like structure which is the fallopian tube. This is a fallopian tube. Various parts are there. The fertilization always occur in the ampulla region. Done. Now, this is one side is clear. Same is there on the another side. Here, ultimately, the fallopian tube opens into this structure. Now, can you see this structure? What is this? This is a uterus. Hystera, uterus is this. Now, this uterus is having various parts. Outermost layer is termed as perimetrium. Middle layer is termed as myometrium. Inner is endometrium. Now, the very important is, what is myometrium? Myometrium is the muscular layer. Muscular. And it is made up of smooth muscles. It is made up of smooth muscles which helps in the contraction. So, here the another layer is the endometrium, right? Endometrium periodic shedding leads to the menstrual cycle. Periodic shedding. That means every month the wall which breaks is the endometrium. Myometrium is the one which structure whose mild contraction helps in the removal of the endometrium at the time of menstrual cycle, whereas heavy contraction at the time of delivery, that is a parturition, is important for the easy birth of the baby. So this is a muscular layer, smooth muscular layer, not under our control. Ultimately, this opens into a cervix. This is a cervical canal. This is a vagina. These structures, they form the birth canal. Cervix and vagina both forms the birth canal. Ultimately, it opens into a birth canal. Done. Now, let's come to this structure. Diagram is given over here. This is the external genitalia one. But I know with this diagram, you won't be able to understand what is the external genitalia. Now, let's have a look. So, don't worry, I'll explain you this diagram also. Just have some patience. So this is how it look like. Here the hairs are present. Hairs. Right? This is the structure. I'll show you. What is this? This is a cushiony structure which is termed as mons pubis. Mons pubis. Here, mons pubis. This is the labia, lips are present, labia majora. On here, hairs are also present here. Now next is labia minora. Labia minora. Now, here the urethral orifice is there. That means here the urethra opens. Here, the next is the vaginal opening is there. This is the vaginal orifice. Here a membrane is present, which is vascular membrane, which is the hymen. So, hymen is not the one which signifies the what do we call that word? I just forgot. That the person, he or she, like the girl, she has earlier she was involved in any sexual act or not. It is not the case. Many times in the horse riding, during the strenuous exercising, during the power yoga, this breaks up. So it is not the case. Vaginal orifice, and this is the hymen present. Clear? Here, here some glands are present. Bartholin gland. Some glands are present which are termed as Bartholin gland. Bartholin gland. Here the whole structure is vestibule. So here the periurethral glands are present. Peri means outside, urethral glands are present. So ultimately these two secrete mucus which helps in the lubrication. Done. The structure is done. So hairs will be present throughout the region in the labia majora and the mons pubis. Done. Let's move on to the next part, that is a mammary gland, and then we will be doing the structure which is the what is left, the ovary diagram, because ovary diagram I want to do with the your menstrual cycle. Now, what is a mammary gland? Mammary gland are paired structure which are also termed as a breast. So their actual development starts at the puberty. So at puberty there is a hormone which is termed as estrogen. Estrogen is the one which helps in the formation of breast, right? So their proper development starts at the age of puberty, and after 20 year of age, in a proper adult, the proper development of breast is seen. So listen very carefully. So here the glandular structures are present, and the size they vary because of the amount of the fat. If fat is more, the size will be big. Glandular tissues are present in each breast, that is divided into 15 to 20 mammary lobes, which contain the cluster of cells which are termed as the alveoli. So look at this structure. I'll explain you everything. Here, look at this. So if you look at the overall structure, here the muscles are present which are termed as pectoralis muscle. What are these cut structure? These are the ribs, and there are the muscles which are present in between the ribs. Hope this structure is clear. Now, here, these are the mammary lobes present. What are these? What is this labeled? C. These are the mammary lobes which are present. What are these? Mammary lobes. Now in this mammary lobes, you will find alveoli are there, right? Alveoli, mammary alveoli are there. So in this alveoli, there is formation of milk. And when does the milk formation occur? Milk formation occur under the action of a hormone which is prolactin, and this prolactin comes from the pituitary, basically the anterior pituitary, which helps in the formation of milk. And this occurs at the time of pregnancy. During the later part of the third trimester of the pregnancy, this starts occurring. Many times it is a positive reflex that after the birth also it happens. So that I'll explain you later on. Formation of milk. Now, this is also under the control of HPL, human placental lactogen, right, which is secreted by the placenta. That means at the time of pregnancy, at the time of delivery, the formation of milk occur in the alveoli. Yeah. Ultimately, alveoli, they are present here, they are present in the mammary lobes. Some lobes are there. So in these lobes, the alveoli are present where the synthesis of milk occur. This is what your first line explains. See, I am explaining you each and every line there in NCERT with the help of the diagram. Now, the cells of alveoli secrete milk, which is stored in the cavities of the alveoli. Clear? So once it is synthesized, it will be stored. The alveoli open into mammary tubules. So, now, this is the alveoli. Alveoli open into the mammary tubules. Mammary tubules. Clear? Is this clear? Now this is one tubule. Like this, many tubules are there. All of them, they join together, and what they form is the mammary duct. What they form is the mammary duct. Right? Let's do the NCERT reading. Alveoli opens into mammary tubules. See, alveoli. This is alveoli. This is the alveoli. Look at this. Right? Alveoli are present in mammary lobes. Alveoli opens into the mammary tubules. Tubules further opens into the tubules opens into the mammary duct. Ultimately opens into the mammary duct. Done? Point clear? No. This is the one mammary duct. This is the another mammary duct. This is the third mammary duct. Ultimately, this opens into a wider region which is termed as ampulla. Which is termed as, look at this. This is a wider region which is termed as ampulla. Right? Now, ampulla further opens into a lactiferous duct. It opens into the lactiferous duct. Lactiferous ducts to open to the outside. Clear? This is the opening, right? Here, some colored part, brownish part is present, which forms the areola. Right? And this forms the nipple. So ultimately, look at the structure very carefully. Lots of alveoli are present in the mammary lobes. This is one lobe, this is another lobe, third lobe, fourth lobe. You will find 15 to 20 mammary lobes are there. In mammary lobes, the alveoli are there. Alveoli opens into the mammary tubules. Mammary tubules opens into the mammary duct. Various mammary duct opens into an ampulla, a thick structure ampulla. Ampulla opens into the lactiferous duct. Lactiferous duct open to the outside and forms a nipple. Look at this structure now. See, mammary lobes are present, in alveoli are present. These are the mammary alveoli which are given. Alveolus done. This opens into the mammary duct. Ultimately, it opens into the mammary duct. Then to the ampulla. This is the ampulla. Ultimately, lactiferous duct to the nipple. Now, brown portion is termed as areola. That's it. The structure is super duper easy. Clear? Come to the next diagram. What is this? The ovaries. Now we have to talk about, one second, let me check the battery level. So okay. Okay, yes. So next is the ovaries. How many ovaries are there? There are pair of ovaries are present. Yeah. There are pairs of ovaries. Now this is one ovary, like this another ovary are present. Now if you look at the ovary, here the outermost is the cortex part. Outermost part is termed as cortex. Here the center one is termed as medulla. Like that of the kidney, the same structure, outer cortex and inner medulla is present. Now if you look at this, the layer here, this is termed as layer. Similarly, germinal epithelium is present. Germinal epithelium. Layer is germinal. Simple cuboidal epithelium are present. When I was there, I'll look at, just go deep into the story. When I was there in my mother's womb, right? Mother's womb. At that time, these follicles were present. Your NCERT says couple millions of these follicles, they were present in each ovary. Millions of these. Yeah, they were present in each ovary. These are termed as primary follicle. Ultimately, they grow and they form the tertiary follicle. They form the tertiary follicle. Ultimately, tertiary follicle form the Graafian follicle. I'll explain you all the structures. If the structure is done, so that means you know everything about it. It forms a girlfriend, which is a Graafian follicle. This ruptures each month. So one follicle grows and it forms the ovum. Every month, one like this will occur. So another follicle will grow next month. Another follicle will grow next month. So whatever which is left behind is termed as corpus luteum. Yeah. If egg is released in the cavity, if ovum is released in the cavity, the one which is left behind is termed as the corpus luteum. Now this corpus luteum acts as a temporary endocrine gland. It secretes hormones such as progesterone and estrogen. This regresses itself and it forms the structure which is termed as corpus albicans. Corpus albicans. This is the corpus luteum. Second one is a corpus albicans. Now let's have a look. This is the primary follicle. This is the primary follicle. This is the primary follicle. Here, it is having the structure which is the primary oocyte. Primary oocyte. Now this forms, this one further develops and it forms the secondary oocyte. So secondary oocyte looks like this with the more follicle cells. Right? So this is termed as secondary follicle. What do we call this? Secondary follicle. Clear? Is this point clear, buddies? What do we have? They have a primary oocyte. What do they have is primary oocyte. Moving on to the next is, it forms the secondary oocyte. Secondary oocyte further forms, this is the structure. It forms a structure like this, which is termed as tertiary follicle. I'll explain you everything. Tertiary follicle. Right? Here the outermost layer that it forms, that is a thecal layer. That's it. Thecal. Right? Here, if you look at the structure, here ZP, that is the zona pellucida starts. Zona pellucida, which is a acellular layer. acellular layer. Clear? It is made up of glycoprotein. The outermost layer further develops into the two layers. One is a theca externa and second is a theca interna. Two layer. Theca externa. See, from the name itself, you can easily derive theca externa. Second is theca interna. So these layers they starts forming. Yeah. What do they get is antrum. Antrum is the cavity. Cavity starts forming. Ultimately, here, what is present is here the primary oocyte. Here the secondary oocyte is there. Secondary oocyte is there. Clear? Now, this one forms the structure like this. What is this? This is again same. Here the cavity size, the antrum. What is this? This is again zona pellucida, ZP. These are the two thecal layers which are present. This is the antrum. And here also, buddies, the secondary oocyte is present. Secondary oocyte is present. Done? This is a structure of a Graafian follicle. Now I'll explain you each and everything. Girlfriend, I'm right. Girlfriend, it's not girlfriend, it's a Graafian follicle. Clear? Now listen very carefully. When I was there in my mother's womb, I was having primary follicle in my ovaries. Millions of these were present. When I was 4 to 5 months of gestation, I was in mother's womb. Now these follicles, they stop developing. They do not develop further. So their number remains the same. Okay? Now at the puberty, like once, that means later on, I was born. And then when I reached the puberty, right, the number of the primary follicle, which were couple million, now they are 60,000 to 80,000 as per your NCR. How many? 60,000 to 80,000. Out of this, one forms the secondary follicle. What does it form? A secondary follicle containing the thecal layer, and it is having the primary oocyte only. These follicles grow because at my puberty, listen very carefully, at my puberty, the FSH level increases. FSH is follicle stimulating hormone. So because of the FSH, this occurs. Here, done. FSH, this grows. Follicular stimulating hormone, the follicles will grow. Ultimately, this egg, this is a primary oocyte. It forms a secondary oocyte with the help of a process which is oogenesis. These are the gametes. The one which is there in the center, these are the gametes. So the one thecal layers or the different ZP layers, these are the friends. You can say this is the main person, and these are the friends which surrounds her. So here the different layers start forming, which is termed as tertiary follicle. Ultimately, the antrum increases and it forms the another, that is a Graafian follicle. Listen very carefully. All these development are under the action of a follicular stimulating hormone. Growing follicle, as the follicles are growing follicles releases a hormone which is termed as estrogen. Estrogen will cause anterior pituitary to secrete more LH. Because of the secretion of the LH, luteinizing hormone, this happens, which is termed as ovulation. Whatever is left behind act as a temporary endocrine body, which is a corpus luteum, and what we will get later on, it regresses itself and it forms a corpus albicans. Clear? The structure clear? Now, let's move on further. Now we have to talk about the topic which is a gametogenesis. Clear? So first let's talk about the oogenesis. oogenesis. Oogenesis is a journey. Oogenesis is a journey which include your folliculogenesis also. Folliculogenesis, that means these steps, they are simultaneous. Follicular genesis, and it also leads to development of egg. First step is folliculogenesis. Listen very carefully. First, I'll explain you this. So this is the same way you have to write in your paper. First, you're going to explain what is oogenesis. Oogenesis involves folliculogenesis, that is a formation of follicle, and it forms a development of egg also. Clear? Now, the next point which you're going to write is, when does it start? These starts before birth. Before birth. That means before the birth of a girl child, this starts. When does it stops? Yes, it stops. It stops at the age of, which is termed as menarche. That is almost at the age of 50 years, it stops. It doesn't go further, 50 years. That means in some ladies it is 45 also, in some ladies it is 55 also, on an average it is 50 years. First, we are talking about the folliculogenesis. So, folliculogenesis means primary follicle converted into secondary follicle, secondary converted into the tertiary follicle, tertiary converted into the Graafian follicle. Clear? Right? What is their number? Their number is couple million. Couple million. Right? Now, as the FSH increases, this process goes on. This process goes on. Follicle formation. That means formation of the friends. F means follicle, F means follicular stimulating hormone, F means friends. Thecal layer, this layer, that layer, that's it. Done? Clear? What is the development of egg? So, development of egg means conversion of primary oocyte into secondary oocyte, and ultimately development of ovum. This whole process of development of egg, it's not a continuous process. It's a it halts at various stages. Now let's understand this. Come to this diagram. Now look at this. Look at this. First, germinal epithelium. From the germinal epithelium of ovaries, listen very carefully, from the germinal epithelium of ovary, some cells starts forming the oogonia. They start forming the oogonia. They become swollen. They become swollen. That is their, you can say they differentiate themselves, and what they form is the oogonia. Oogonia undergo the mitosis, and what they form is a primary oocyte. It's the primary oocyte. What they form is a primary oocyte. So what they have started doing, the mitosis, what they form is a primary oocyte. Right? Primary oocyte. So when I was there in my mother's womb, so at that time, oogonia were there. Oogonia started multiplicating themselves, and what they formed is a primary oocyte. Primary oocyte with the help of a process which is mitosis. With the help of a process which is mitosis. So once the mitosis, primary oocyte. So primary oocytes, they were millions. They were millions. Now these primary oocyte, all the millions of primary oocyte, they started undergoing the meiosis. Now let's look at the story. Now they want to go into a journey. They started undergoing the meiosis. As they entered into the meiosis, first halt occurred. That is a prophase 1. So all these, they were halted. So here, one second, here the halt happened at prophase 1. That is the diplotene stage, right? So first halt happened, that is a diplotene stage. So it is like, listen very carefully. Oogonia divide and it forms the primary oocyte. What they formed is a primary oocyte. Now primary oocyte said, because I was diploid, the primary oocyte said that I want to complete meiosis. Ultimately, what does it form? It forms a gamete. So what they want is, they want to undergo the meiosis. So it is like, this is a boundary. They entered in the meiosis. As they entered in the meiosis, they were halted. So they halted at the, see, first stage is prophase only. So they halted at the prophase 1 stage. So that means all the primary follicles, they were at the primary oocyte stage. Primary oocyte, that means they were halted at the prophase 1 diploty. Then they remain like this for 14 years. Let's say they remain like this for the 14 years. 14 years means, that means till the age of puberty. Normally, what do we say? Puberty occurs at the age of 14. And you know, because of adulteration and because of so many things today. So it is like, you won't believe, there are people, there are girls who are at the age of 9 years old. They are getting menstrual cycle. So let's not go into that topic. So primary oocyte. So they are halted at a stage of prophase 1. So they remain in that thing for 14 years, like till puberty. Then puberty, at puberty, again, there is a first role of follicular stimulating hormone. Follicular stimulating hormone came. Follicular stimulating hormone started forming follicles. Clear? Follicular stimulating hormone started forming the follicles. Follicles start, that means friends started forming outside it. Friends started forming outside. They started secreting estrogen. They started, listen very carefully, they started secreting estrogen. Because friends, that is a follicles, they started secreting estrogen. That triggers, due to which the estrogen, which is responsible for the secondary sexual characteristics, now these prophase 1, which were stuck at the prophase 1, out of this, one started undergoing the meiosis. So they completed the meiosis 1. So what, till puberty, at puberty, they have completed the meiosis 1. So what they formed is the secondary oocyte. Secondary oocyte. That means primary follicle, secondary follicle, and now the tertiary follicle. Secondary oocyte, tertiary follicle. Look at this diagram. Secondary oocyte, this is the tertiary follicle. Secondary oocyte. Yeah. Now, at the once she got her my first at the puberty, so this happened. Secondary oocyte further wants to, as the first step, meiosis 1 completed. So meiosis 1, if you remember, if one cell meiosis occur, what do we get is, the first division is a reductional division, right? So the number of the chromosome, they will be reduced to half. So initially, when it was 46, their number will be reduced to 23. So what we will get is a 2 cell. One is a bigger cell, which is a secondary oocyte containing most of the cytoplasm. Another small cell, just a nucleus, that is termed as a first polar body, which is of no use to us. Then after meiosis 1, what happens is the meiosis 2. So meiosis 2, when it happens, here there is a formation, it has to formation of the egg. But this is the second halt. At meiosis 2, metaphase 2, they are halted. They are halted. And as they are halted, secondary oocyte, as they are halted, they are pushed from the ovary. Now this egg will come outside in the uterine cavity, outside in the cavity. They will be taken up by the sperm. Oh, sorry, they will be taken up by the fallopian tube. Fallopian tube, in fallopian tube, they will reach to the ampulla region. And once they will reach the ampulla region, in ampulla region, they will wait for the sperm. If sperm will occur, they will complete the meiosis 2. Are you getting this point? If sperm will occur, they will complete the meiosis 2. If sperm will not occur, then they will die. That means the menstrual flow. Done? So everything is under the control of the hormone. Now let's understand this again. This is a oogonia, right? It forms a primary oocyte. I was at this age of my, now listen very carefully. I was at the age of my 5 month. So there were lots of primary follicles containing the primary oocyte was present in my each ovary. It remained like this. They were arrested at the prophase 1 diplotein stage. It remained like this. When I got my first menstrual cycle, which is around the 14 years of age, what did I don't remember also. So at that particular time, Now these follicles, they started developing. Follicles, primary follicles form the secondary follicle. Secondary, tertiary ultimately forms the graphite follicle. This is the follicular genesis. Now the next is the oogenesis, that is development of egg will also occur. So here, what will happen? The one which was arrested at my 14 year of age, the one which was arrested at the prophase 1 stage, it will complete the meiosis 1. There will be formation of the one polar body. It will start undergoing the meiosis 2, but they will be arrested at the metaphase 2. Now my ovary kicked that secondary oocyte which was arrested at the metaphase 2, and that entered in the ampulla. That one waited for the sperm to occur. It has a life of around 24 hours. It waited for 24 hours for the sperm to occur. If no copulation, if egg was not fertilized, what will happen? The egg will go away. As the egg will go away, so menstrual flow will start. So how does it occur? That is also under the control of the hormone that I'll show you in the menstrual cycle. So hope this cycle is clear. Now this second very important question, when does the second polar body formation occur? Second polar body formation occurs at the time of sperm penetration. Clear? First division occur at the age of meiosis 1 occur at the age of puberty. Clear? So when I was in mother's womb, this was arrested at the prophase 1 stage. So it waited for around 14 years. Clear? So initially the cells was 46 only. Once they'll complete the meiosis 1, they will become 23, and later on they will remain the 23 only. Yeah. Moving on to the next point, that is a spermatogenesis. So what is a spermatogenesis? Spermatogenesis is the formation of sperm. Spermatogenesis is the formation of sperm. Now, let's understand from this diagram only. Spermatogenesis. So, listen very carefully. There were germinal epithelium. There was a germinal epithelium. Germinal epithelium forms the, out of this, some germinal epithelium, some layer of the germinal epithelium, it was also a simple cuboidal kind of cells. They growing further, they form the spermatogonia. Spermatogonium. What they form is spermatogonium. See, understanding this topic is important. If you are able to understand, that means you will be able to write down anything. Clear? I'm not here to give you notes. If I started giving you notes, then it would have taken 5 to 6 hours of session. It's a summary series, so just try to understand this topic. If you will be able to understand, that means you can write the things in a better way. Spermatogonium, it forms the, for spermatogoniums, whatever was there, it forms a primary spermatocyte. Primary spermatocyte. With the help of a process which is termed as mitosis. Clear? Now the next is, buddies, look at this. Now these primary spermatocytes, it forms the secondary spermatocyte. Secondary spermatocyte. What does it form? Secondary spermatocyte. Secondary spermatocytes, here meiosis 1. Meiosis 1. Now it started forming spermatid. Rounded. Clear? Meiosis 2. Meiosis 2. This spermatid forms the sperm. Yeah. Now let's understand what is this spermatogenesis. Spermatogenesis is the process of formation of sperm. That means a journey from spermatogonium to sperm. So this whole journey is termed as spermatogenesis. We are making you understand the topic. Now the next point is, when does it start? When does it start? It starts at the puberty and it never ends. It continues. Clear? Clear? This is under the action of testosterone that we have discussed. Now, germinal epithelium, spermatogonium start forming, which undergoes the process of mitosis. Right? So here the cells were 2n only. This was also a 2n. They were undergoing the mitosis. They were also 2n. Yeah. Now, there was a one primary spermatocyte. One primary spermatocyte. One primary spermatocyte formed two cells which are termed as the secondary spermatocytes. Clear? These two, because they were the reductional division part, so they were N. Now the next is this one further develops into the spermatid. Now these were also N. Clear? And this started forming the sperm, and this is termed as differentiation. Now differentiation, buddies, here mean, here the cells, if you look at the cells, spermatids, they are round. But what's the structure of sperm? Sperm structure is like this. Structure is like this. Yeah, so this is how the sperm. Here, at this particular time, from spermatid to sperm, there are lots of things that happened. Like the cytoplasm reduced, the Golgi body started forming the acrosome, and they attained this shape. Now, this particular process, that is the formation of spermatid, formation of sperm from spermatid, is termed as spermiogenesis. Spermiogenesis. And if you look at these sperms, they are embedded in the Sertoli cells. These are the Sertoli cells. Sertoli cells. So they just come out from the Sertoli cell. They come out from the Sertoli cell. They come out from the Sertoli cell. Now this process is termed as permeation. Now there is a difference. What is a spermatogenesis? Whole procedure is spermatogenesis. What is a spermiogenesis? Spermatid to sperm. What is spermiation? That is Sertoli cell, because they were embedded in the head of Sertoli cells. They are released now. Now this will be termed as the spermiation. Process will be termed as spermiation. Clear? So look at this diagram. Spermatogonia, puberty. It starts at puberty only. Puberty, mitosis, differentiation, primary spermatocyte, everything I have written. Differentiation. So this is not your mathematics wali differentiation. This differentiation is completely different. Here, that means specialization. Clear? Now, moving on to the next part, which is a menstrual cycle. The menstrual cycle, it means regular, or let's say monthly formation and breakdown of endometrium lining. Endometrium is your uterine lining. What is this? This is your uterine lining. Right? So this is the cycle which occurs in the uterus. This is not the case. This is not the only case. Next is development of follicles. Second step is development of follicles. In development of follicles, where does the follicle development occur? In ovary. So these are the things which occurs. We cannot say that the process only occurs. See, listen very carefully. We cannot say that this particular process only occurs in the endometrium. It occurs in the ovary also. So steps happens simultaneously. Now, first step, let's talk about the one which happens in the uterus. Clear? So here, listen very carefully. As the progesterone level reduces, estrogen reduces. The first step is termed as a menstrual flow phase. So the endometrium, in this case, the endometrium falls off. Blood comes out. Here, the blood comes out from the female body. This will be termed as a menstruation. Clear? And this is total 5 days. How many days? 5 days. I'm talking about the uterus. Okay? Now the next step, listen very carefully. Next step in the uterus, just mark it, guys. Second step in the uterus is a proliferation phase. I'm saying proliferation. Now I'm not saying the follicular phase. This is a proliferation phase. Proliferation phase, that means formation of endometrium. Formation of endometrium. Because of the increase in the estrogen, proliferation of endometrium occur. Proliferation of endometrium occur. Clear? Now the next third one is termed as the secretory phase. Telling you with respect to the uterus. Okay. So this is the secretory phase. Now here, because of the increase in the progesterone, the secretory nature of the endometrium starts occurring. Secretory nature. Secretory nature, that means now they are more glandular. They will be secreting some things which are important for the baby development. Done. Here, this is with respect to the the uterus. First step, menstrual flow phase. Second step, proliferation. Third step is the your secretory phase. Done. Clear? Let's talk about the same process. Means simultaneously, parallelly, the same process, not the same. At the same time, the same thing happens. Kind of things happens in the ovary also. So in ovary, these follicles, they starts developing. Primary starts forming secondary. Secondary forms the tertiary. Tertiary forms the girlfriend. That is a Graafian follicle. Graafian follicle. Because of increasing level of FSH. See, FSH is there. FSH level is there. The follicles, they start forming. Developing follicles, listen very carefully. Developing follicles starts forming estrogen. Estrogen will cause LH to increase. So LH will cause LH surge. LH surge. This will be termed as ovulation. So the first stage in the ovary was the follicular phase. So here I am writing. First step in the ovary was follicular phase. Follicular. Here, increasing FSH will cause the development of the follicles, due to which the estrogen level increases. Second step is the luteal phase. Second step is termed as ovulation. Ovulation. Increase in the estrogen will lead to increase in the LH. LH surge, ovulation occurs. See, whatever is left behind is termed as corpus luteum. So the next step, the step number 3, is the luteal phase. Luteal phase, that means corpus luteum formation. Now corpus luteum is a temporary endocrine gland. This forms the progesterone and this forms the estrogen also. Clear? As it has a life, it's a temporary. As it regresses, see, the level will go down. As the level will go down, the menstrual flow starts. So this whole cycle is of 28 days on an average. It varies from person to person. And if there is 3 to 4 times in a year, there is drastic change in the menstrual cycle, one should go and see a doctor. Clear? So it's a 28 days cycle. In uterus, first thing is menstrual flow phase. Second is proliferative, third is secretory phase. In the ovary, there is follicular phase, there is ovulatory phase, and there is luteal. Done here. So hope this diagram, the flow diagram, a very important, very easy diagram, is clear to you. Done? Happy? Okay. Next step is fertilization and implantation. See? We are revising everything. Are you enjoying? Yes? Okay. So, fertilization. So, what does a fertilization do? Fertilization here, first thing is there will be sperms. So, sperms, they are deposited into the female reproductive tract. 1 second. Okay. Sperms are deposited into the female reproductive tract. Once sperms are deposited, insemination occurs. Clear? Now, the sperms, they started penetrating. Now, first step is the acrosomal reaction. acrosomal reaction. In the acrosomal reaction, the enzymes which are there in the acrosome found by the Golgi, the enzymes, they will be released. So the name of these enzymes are, listen very carefully. Listen very carefully. Now, the first is termed as hyaluronidase. Hyaluronidase. Where is hyaluronidase? What will hyaluronidase do? Here, if you look at the follicle cells, they are attached to each other with the help of the hyaluronic acid. Mucopolysaccharide. Hyaluronic acid. Hyaluronic acid will be digested by the hyaluronidase. Clear? Second is corona, your favorite, corona penetrating enzyme. Corona penetrating enzyme. Now, where is this corona present? These are the cells of corona radiata. Corona radiata cells will be digested by the corona penetrating enzyme. Third is the zona lysine. Zona lysine. Now, zona lysine will digest the zona pellucida. Clear? Acellular layer. Now, these are the enzymes which are secreted by the sperms. Acrosomal reaction. Second thing is, there is a step number 2 also. That is the cortical reaction. Not given in your syllabus, but it's okay. So, cortical reaction is that reaction by which it prevents polyspermy. So what happens is, cortical granules, they fuses in the perivitelline space, due to which if one sperm is entered, other sperms, they will not be allowed because there is a cortical reaction. Clear? In perivitelline space, the cortical granules, they will show exocytosis. Okay, and there is also a species specific thing. It's not the case any sperm from another species, they can fertilize the egg. Done. Move on to the next. The first step is the formation of a zygote. Right? See, this is the egg. What is this happening? Fertilization. See, only one sperm has entered. Other sperms, they are not allowed. So that means here polyspermy is prevented. Here the zygote will form the 2-cell stage. Here the division is mitosis, a kind of mitosis division, but this is termed as cleavage. Because the daughter cells here, because the blastomere, here we do not say the daughter cells, mitosis, it's like a mitosis only. It is a kind of mitosis, but there is a difference. The first thing is they are not termed as daughter cell, they are termed as blastomeres. They will form the blastomeres. And whenever they develop further, the cells, the daughter cells, the blastomeres which are formed, blastomeres, they are of smaller size as compared to the parent one. In mitosis, blastomere, they are of equal size. Clear? Right? In normal mitosis, they are of equal size. 1 second. Clear? They will develop further, 4-cell stage. Ultimately, they will form a 8 to 16 cell stage, which is termed as morula, mulberry. Right? Mulberry, morula. Clear? Now, this is how the development occurs. Ultimately, they start forming the blastocyst. What's a blastocyst? Now, the cells, they start arranging themselves. The outermost layer will be termed as trophoblast or the trophoactodum. Trophoblast or the trophoactodum. This will be termed as ICM, inner cell mass. Inner cell mass. Now, trophoblast forms the placenta and next embryonic membrane. Placenta, it forms the embryo. It forms the embryo. Done? Clear, everyone? So, ultimately, here they will implant itself at the 7th day, 5 to 7 days. Ultimately, implantation occur. Clear? So, implantation will occur. The baby will develop for 9 months. The next stage will be the gastrulation, where the 3 germ layer will be formed: ectoderm, endoderm, and the mesoderm. Ectoderm form the various structure like nervous system, the skin, outermost structures will be formed by the ectoderm. Mesodermal, that means blood. Endodermal means your gut, your liver, your kidney, they will be formed. Done. So look at this. Look at this. Very, very, very important. Here. This is the embryo. This is the structure is termed as embryo. What's the name of the structure? Embryo. Embryo develops from inner cell mass. Embryo develops from inner cell mass. Clear? Now, next structure, you can see this one. What is this? This is the umbilical cord. This is the umbilical cord by which the placenta is attached. Here, so placenta, this is a placenta, and placenta remains like this in the uterine epithelium. So this is a uterine wall, right, which are having crypts. This is a uterus which are having crypts, whereas the placenta remain like this. This is the placenta. This is umbilical cord. Now this is the baby. Clear? So these are the villi, chorionic villi-like structure. Clear? Now this placenta is formed by the trophoectoderm or the trophoblast. So they are formed like this. They are present like this. Umbilical cord is the one which is connected. Done? Are you getting this point? So initially there is a formation of placenta which provide nutrition. Later on umbilical cord starts forming from the placenta. This is the embryo. Done? This structure is clear? Yeah. There is one more structure which is termed as yolk sac. Yolk sac. So yolk sac is that region where the RBC starts forming first. Though it is reduced, but it has a function of forming. Listen very carefully. It forms the RBC. You know, the first structure which is formed in the baby is a heart. Heart is a pumping structure. If a pumping structure is there, so that means a blood should be there. So blood formation, RBC formation, occurs in the yolk sac. Erythropoiesis, when a baby is there in the mother's womb, its formation occurs in the yolk sac. Later on, yes, its development occurs in spleen also. Then their development occurs, see, first their development, listen very carefully. It occurs in the yolk sac, then the liver, then the spleen, then ultimately the long bones. So in our case, long bones. When I was there in mother's womb, it was first the yolk sac, and then it was the liver, then it was the spleen, and then it was the long bones where the erythropoiesis occur. Clear? This point is clear. So there is a cavity of the uterus also. You know, the baby, this is a baby, right? Here, this the amniotic fluid will be present. You know, many times at the time of delivery, we say water bag is burst. So what is that water bag? That's a fluid containing bag where the baby is floating. Clear? Where the baby is floating. Done? Okay. Guys, just give me 1 second. I have to change the batteries and then we will proceed because there are a lot of things left. Hello, hello, hello. Hope I'm audible. Yes, great. Clear? So this is how the structure look like. Here the amniotic fluid is present. Clear? Amnion, chorion, as the structures are not there in your syllabus, so we are not focusing in that. Now, next is development. Development is very important. 1 month of pregnancy, first structure which is formed is the heart. This is what we make heart. So the first structure which is formed is the heart. Next is the first growing fetus may be by the heart sound through the stethoscope, and in fact it's nowadays it is like dhak 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 sound. So with the help of vaginal ultrasound, one can listen to the sound. By the end of the 2nd month of the pregnancy, fetus start forming limbs and the digits. Second, limbs and the digit. Next, here The most of the organ system, that is the first trimester, all the organ system, they will be formed. Limbs, external genitalia, organs are well developed. First movement is always seen at the 5th month. So 5th month. So these are the milestone. If you will be a gynecologist, there you will be looking at all these symptoms, like if these symptoms they are occurring properly or not. You have to go through them. Okay, if it is there, that's great. If it is there, that's great. That means the proper development is there. Clear? By the end of 24th, second trimester, 24th week, the body is covered with a fine hair. Eyelids, eyelashes starts forming. By the end of the 9th month, the fetus is fully developed and ready for the delivery. Clear? Next is parturition. Parturition. So parturition, listen very carefully. Here, hypothalamus is the structure. Hypothalamus. First comes the fetal ejection reflex. Fetal ejection reflex. This comes from the developing baby, and developing baby, when a baby is there, and placenta. This gives a signal to the hypothalamus. Hypothalamus will tell posterior pituitary. This will secrete oxytocin. This will secrete oxytocin. Oxytocin will do the two function. Strong uterine contraction. Strong uterine contraction. It will cause strong uterine contraction. It will also cause milk ejection. Milk ejection. Here, milk synthesis is a prolactin, but milk ejection. Clear? Are you getting this point? So, milk ejection. Now, more and more strong uterine contraction, more signal will go to the hypothalamus. More posterior pituitary will start secreting more oxytocin. More and more uterine contraction will occur. Ultimately, baby will enter into the birth canal and the easy delivery can happen. Now, what are these things which will cause? Not only this, there are some prostaglandins also which are released, which causes the easy birth of the baby, but not given in your syllabus. So, we'll not go into that detail. Now, baby and the placenta, both of them, they start the fetal ejection reflex. Done. Hypothalamus, strong uterine. Acha, I was telling you that milk synthesis is because of the prolactin. Milk ejection is because of the this, right? Now, very important part is the placenta. Placenta is also a temporary endocrine gland. It is also a temporary endocrine gland. You know, it is also a temporary endocrine gland. You know, what does it secrete? Very, very, very important. The first hormone is HC, that is the human chorionic gonadotropin. And this HCG measurement is there in the pregnancy kit. This is measured in pregnancy kit, whether it is positive or negative. If HCG is there, that means placenta is there, that means positive pregnancy. Second is human placental. This I have discussed, human placental lactogen. Now, placenta also secretes your estrogen. It also secretes because there is a life of a corpus luteum, and for maintenance of pregnancy, the progesterone is required. So, progesterone is also secreted. Now, the next is, there are some hormones like cortisol level also increases, not secreted by the placenta. Cortisol level also increases in pregnancy, and thyroxine level also increases to maintain the higher metabolic rate. Also increase. You know why? Because baby is considered as, please don't mind this, baby when it is there in the mother's womb, it is considered as a parasite. So, baby takes everything from the mother. So, it is like a parasite is there in the body and which is taking away everything. The body has to maintain that. Are you getting that point? Mother has to maintain everything, so that extra should be provided to the baby. high metabolic rate. So at that time, protein content also one has to increase. Not only the protein, the fat also one has to increase. The carbohydrates also, see, good carbs in fact. Yeah, that we should increase. Now, done? Now, let's do some MCQs, guys. How many autosome? So, this was all about the whole chapter where we have talked about lot of things from first topic to the last topic. Now, let's do some questions and I'll give you some homework also. Now, you are going to tell me the answer in the comment section. This is the first answer question, guys. Read this question. How many autosome does a human? See, these type of MCQs, they will be asked in your paper. And questions, they will be super easy. Autosomes does a human primary spermatocyte have? Primary spermatocyte, how many will be there? There will be, autosome will be 44, total is 46, but autosome will be 44 only. They will be diploid. Now, after ovulation, Graafian follicle regresses into corpus luteum. This will be the right answer. Mature Graafian follicle is generally present in the ovary of a healthy human female around. Mature Graafian follicle. Where it is present? 5 to 8 days? No. 11 to 17? Correct answer is this. Mature Graafian follicle. Mature girlfriend. Now this answer you are going to give me in comment section. Which of the following is not a male accessory gland? This is your homework, guys. I'll be waiting for your answer in the comment section. Do answer, okay? Let's look at some short answer type question also. Mention the relationship between the pituitary and ovarian hormone in the menstrual cycle. This was a PYQ. So pituitary hormone, that means you have to talk about the role of FSH and LH. Ovarian hormone, that means you have to talk about the role of the estrogen and progesterone. Estrogen and progesterone. Clear? Are you getting my point, buddies? These two you have to talk about. Clear? Short answer type question, 3 marks at least you have to. Now, name the stage of human embryo that gets implanted into the uterus and draw its label diagram. What are you going to draw? Blastocyst. 3 marks, well labeled diagram. Inner cell mass, the trophoblast, trophoectoderm, blastocele cavity, everything you have to draw. Okay. So your diagram should be with pencil. Labeling should be on one side and the image lesion should be there. Like this is what you are drawing. Now, let's look at some long answer type question also. Where do spermatogenesis occur in human testis? Where does it occur? Spermatogenesis in the testis only. Now, human testis, seminiferous tubule. Describe the process of spermatogenesis. Can you do that? So all the stages you have to write down. Trace the path of spermatozoa up to ejaculatory duct only. So from the testis to the tubuli recti, then to the retitestis, vasa efferentia, epididymis, vas deferens, then seminal vesicle duct, and then the ED, ejaculatory duct. Clear? So whenever you are having a questions like this, spermatogenesis, try to don't write, just make a flowchart diagram. Now, moving on to the next is explain the menstrual cycle. Very important, menstrual cycle. See, the question seems super big, but it is very easy. So, menstrual cycle, state the level of ovarian and pituitary hormones. See, this is important. This was the question which was asked in 3 marks also. Why is follicular phase is also termed as proliferative phase? Follicular, that means follicles formation. Proliferative means endometrium formation. Explain the event that occurred in the Graafian follicle at the time of ovulation and thereafter. Draw the diagram of a Graafian follicle. Will you be able to do this? Easy? Now, look at next question. Hormonal regulation of spermatogenesis. FSH, LH. Very easy. Now, next is diagram of a human sperm. Write down the component of its head. What are there? Acrosome is there. Here in the head, the nucleus is also present. Clear? So, hope you were able to understand the chapter. Was it easy? Easy peasy lemon squeezy type? Yes, many times students, they come to me and they say, ma'am, this chapter is complex. No, guys, it is not. It is super easy chapter. Only clear understanding of various topics are required. So take care, guys. See you in the next session with a new chapter. Till then, enjoy yourself. Do complete the homework and do let me know the answer in the comment section. I'll be waiting for your answer. Have a nice day. Bye-bye. Thank you so much.